I'll be talking about 9.3 skeletal muscle contractions. Also, I'll be identifying the major events of skeletal muscle fiber contraction. In this slide, I'll be talking about depolarization and calcium ion release. An action potential from a motor neuron triggers the release of acetylcholine into the motor end plate. Acetylcholine initiates depolarization within the sarcolemma, which is spread through the muscle fiber via T-tubes. Depolarization causes the sarcoplasmic reticulum to release stores of calcium ions. Calcium ions play a pivotal role in initiating muscular contractions. In this slide, I'll be talking about actin and myosin cross bridge formation. On actin, the binding sites for the myosin heads are covered by a blocking complex troponin. Next, the calcium ions bind to the troponin and reconfigure the complex, exposing the bind sites for the myosin heads. The myosin heads then form a cross bridge with the actin fulfillments. I'll be talking about sliding mechanisms of actin and myosin. ATP binds to the myosin head, breaking the cross bridge between actin and myosin. ATP hydrolysis causes the myosin heads to change position and swivel, moving them towards the next actin binding site. The myosin heads bind to the new actin site and return to their original conformation. This reorientation drags the actin along the myosin in a sliding mechanism. The myosin heads move to the actin fulfillments in a similar fashion to the way in which an oar propels a rowboat. Our last slide today, I'll be talking about sarcomere shortening. The repeated reorientation of the myosin heads drags the actin filaments along the length of the myosin. As actin filaments are anchored to Z-lines, the dragging of actin pulls the Z-lines closer together, shortening the sarcomere. As the individual sarcomeres become shorter in length, the muscle fibers as a whole contracts. Thank you so much, and this concludes my PowerPoint presentation.